Right, hi year 11, this is Mr. Lim here again, and this is another video in acids and bases about indicators this time. So let's get started. Right, so we're going to learn what these indicators are, how they work, that kind of stuff. So let's have a look. Acids and bases are all clear colorless solutions. Okay, so acids and bases are both clear colorless solutions, and it's impossible to distinguish from each other, acids and bases, or even from water, just from their appearance. Okay, so you could have acid, a base, and some water in 3 beakers, and you wouldn't be able to tell the difference between them because they're all clear colorless solutions. Right, so to help identify acids and bases and neutral substances, which are just, not acidic or basic, so it's like water, we use substances called indicators. Right, so indicators, they change their color based on the solution that they are within. Right, so in other words, there'll be one color for acids, one color for bases, maybe one color for neutral, but maybe not. Right, there are many different indicators. Actually, so wait, just with that, that they change their color, it's really based on the concentration of hydronium ions. So they'll change their color based on the concentration of hydronium ions. If it's high, it'll be one color, low, another color. But as you can see here, there are many different indicators which have different colors for when they are in acids, bases, and in neutral substances. So the different indicators, and you can see some here, they have different colors based on their pH, right? More importantly, and this is really important, different indicators can change colors at different pH levels. This means that some indicators cannot distinguish between certain concentrations of acids and bases, depending on what the concentration of those indicator changes are. So what does that mean? So let's have a look over here. Here I got some thymol blue. Okay, some thymol blue will be pink, orange, yellow, green, and blue, right? Which is kind of useful, but if you look at this here, if they've got a pH of 7.5 and a pH of 6.5, this being acidic, this being basic, you wouldn't be able to tell the difference because they'd both be yellow at that point in time, right? Nor could you tell the difference between a very basic thing and a slightly less basic thing, right? More like even methyl orange here, you see that like if it was pH 5 or pH 9, it'd still be the same color. So that would not be able to tell the difference between this particular low concentration of acid and this base here, or even this base here. But once you get to the higher concentrations of acids, you'd be able to tell the difference. Okay, so that's what it means by the color change area is different for each indicator, and it can change to be a different, certain ones have certain color changes at different pH ranges, right? So that's the idea there. So if we look at a bigger picture of this one here, the idea is that you'll get questions about what color certain things will be at certain pHs. So if I say that, what color will a certain indicator be at pH 3, right? So you'll be able to use the color chart to be like, okay, well, bromocresol green is yellow at pH 3, but methyl red is pinkish red at pH 3, right? Or bromophenol blue is about going to change between green and yellow, right? Or you might say that, okay, well, what color is these solutions at pH 9? Okay, so pH 9, you'd say, okay, well, bromophenol blue is blue at pH 9, or bromothymol blue is light blue at pH 9, or phenol red is pink at pH 9, right? So that's what it might say is like, okay, you use a color chart to identify what color something will be at a certain pH. Should be able to, good. Then, stupid thing, why would you erase? Anyway, then you can also think about, okay, well, like this phenol red here will be this kind of beigeish color all the way from pH 7 all the way to pH 0, okay? Or you might say that this Congo red will be red all the way from 5 to 14, okay? And so you would not be able to tell the difference between certain acids and bases. So if I say I have a solution of pH 6 and a solution of pH 9, and then I said, okay, well, come on. Hey, why are you not erasing? Okay, so if I have a solution of pH 6 and a solution of pH 9, okay, then you say, right, would Congo red be able to distinguish between pH 6 and pH 9? Right? If you put Congo red into each of these things, right, Congo red here at pH 6, okay, it's going to be red. At pH 9, it's also going to be red. So it wouldn't be able to distinguish between those. So you'd be like, no, Congo red cannot distinguish between these two. Which indicator could? Well, you got a whole bunch to pick from, but you've got to find something that's different color at pH 6 and at pH 9. So you might say, well, phenol red will be able to. So will phenolphthalein, right? Those will be able to pick out something for that, right? That's the kind of questions that you'll get about this. All right, that's it. It's reasonably straightforward, and you just have to use the color chart there. All right, adios.